studying a group of stingrays that occur on the coast of Brazil. And based on genetic and morphological data, we found out that the stingray that used to occur in Brazil actually belonged to a different species. They used to think it was a species with a wide distribution from the north of the United States until the south of Brazil. But then we found out that the one that occurs in Brazil was a different one. So we described it last year, and we combined genetic, morphological, and ecological data and described it. And right after it was described, there was a meeting by the international UCN, the specialists, and they evaluated the species. So it was right on time. We published it, and then they evaluated it, and it was evaluated as vulnerable. So the species, we didn't even know it existed, and it was already under threat. And it's a good thing that this species has one population in an archipelago in the northeast Brazil, where there is a marine park, so it's preserved over there. But it's only a small part of the whole species distribution. It's only on that archipelago and not on the whole Brazilian coast. So now it's time to act on specific management plans for these stingrays, because we have no idea what have been going on with it, especially because in northeastern Brazil, people usually eat stingrays. They like to fish and eat. And they are not only target species, but they are also bycatch. So when they are trawling shrimp and other fishes, usually these stingrays, they come on the nets. And fishermen, they use it, they sell it, or they eat them themselves, or sometimes they just cut their stings and throw the stingrays back on the water. But they need some education. It's not that they do that because they're mean. It's just that because they don't know. So when I was on the boat with them, I tried to explain it. I tried to teach them what I knew and give them some instructions. So maybe that's what it's lacking, some instructions for these people to know which species are threatened and what they should do and what they should not do. And going on with this subject, me and my co-authors, we are also working on other stingrays, the same group, the same genus. And we found out that in a genus that used to have 9 species, there might be 14. So many more lineages than what was previously thought. And given by this example of this stingray that we described last year, that we didn't know it existed, and when we formally described it, it was already vulnerable, we can think about the other lineages that are not formally described, and they can also be under threat. But since they are not described, they're not going to be evaluated. So this is one of my concerns, because based on genetic data, we know that they exist, but we still do not have more data, like morphological and ecological data, to describe the species and to give the data to specialists so they can analyze. So one of the ideas would be for the specialists to use these genetic lineages as a starting point to do the analysis of their threatened status. Because like another species that has a wide distribution, it's going to be split into 2. So the wide distribution is going to be smaller, and if you decrease the area of distribution of a species to an area that's way smaller, maybe this area, has a larger fishing pressure, and the species that's occurring there might be under a larger pressure and under some threat that we're still not aware of. So this is another big deal, and we're trying to address it. I don't know exactly how, but we're raising concerns about it. I think it's a continuous thing. You have to keep telling them, keep teaching and showing, because we are used to doing our analysis at home, at the lab, on our computers, and we don't go back to the field to talk to these people that fish these stingrays. And that's something that I would like to have done, and maybe in the future I can go back to these places where I went and tell them, look, this stingray that you fished here and you gave me a tiny sample, it's endangered, not endangered, but it's under threat. It's a vulnerable species. I'd like to tell them that. There are lots of people, at least that I'm aware of in northeastern Brazil, that are doing it. So they keep going to fish markets and they keep talking to fishermen. There are NGOs that do that. And I think they're getting good results, but there's still a lot of work to do. And not only in one place, but the whole coast. Because if we preserve one species only in one city, it's not enough based on its huge geographical distribution. We need to do something encompassing the whole distribution, not only one tiny spot. They can be more aware of the species that are there. So for example, these stingrays that are under threat, they could no longer target these species because that's something that they sometimes do. So they can avoid doing that. Another thing would be to release them. When these stingrays are caught, they can survive for a short period out of the water. So when they catch them, they could just, I don't know, that's something for the fishermen because they would need to cut their nets. That's why they'd rather cut the sting of the stingray instead of cutting the net, because the stings are very dangerous and they can hurt very badly. So fishermen have to choose if they want to remove the sting or to cut their net. That's something that we can teach them how to deal with it, how to handle the stings, how to use a glove to remove the net from around the sting. It's something that can be taught and they can learn it. We just need some people to go there and do it. Fortunately, there are some NGOs, but not that many. In Brazil, there is a law, and there is a law with a list of species of fish species that are under threat and that cannot be fished or can only be fished during some periods of the year. But this list comes and goes. So like in 1 month, it's valid and fishermen are forbidden to fish. And then the next month comes another politician and says, no, it's no longer valid. And then the next month comes another politician, no, now it's valid. So we never know. For now, I don't know if it's valid or not, because every single month it keeps changing. The list is there. Specialists on fishes from all over Brazil, they have done an extensive list of fishes based on their threatened status, if they can be fished or if they can't, on their population trends, everything. So specialists know, and they made a list. But if this list is gonna be followed and if fishermen are gonna respect that, it's another issue because we need fiscalization. And there's no fiscalization, so they can do whatever they want. Recently, a paper came out that some researchers from Rio de Janeiro, they did a research on fish markets over there. And they sampled fishes that were being sold. And they took to the lab to examine their DNA and to know which species were being caught. So I don't know, fishermen said it was species A. And when they sequenced, they noticed it was species B. And they were selling species that they were not supposed to be selling.
Wow.
Yeah, because they don't sell the species like the stingray. They don't sell the whole stingray. They cut into pieces, so they remove the head, they remove the tail, and sharks the same. They remove the fins, and you can only see like a cigar of the shark. That's what I say, like a trunk of the shark. So you can't identify the species. Though the person can say it's selling one species of shark, when in fact it's selling a threatened species, and we wouldn't know just by looking. Only with DNA we can know. it's the best way to know, especially nowadays, because we have a huge database of, the barcode gene of pretty much all species, all shark and rays and fish species. We have the barcode of all species. So if we sample only one specimen and we compare to our database, we can know which species that. So the barcode helps a lot in this kind of work. And maybe with money, of course, we need money. The fiscalization could be done this way, like by sampling and sequencing. Of course, we need a lot of money to do that. But that's one way to do it, and to help identify which species are being sold and which ones that should not be there. Yeah. I used to be reluctant to the usage of DNA because my background was in anatomy. So I used to be a comparative anatomist, and I like to study specimens and their musculature, their cartilages, their bones. But when I found out and started studying DNA, it opened my mind a lot, because not only we can use the DNA for these kind of things that we're discussing right now about conservation and species identification using only one gene, which is very simple. We can also use the whole genomes, or not only the whole genomes, but big chunks of the genome to study their evolution, which is also very important. It's, of course, it's more of a basic science. It's not really applied to conservation immedately. But if we understand the evolution of the species, we can understand when they originated and how, why. We can start combining the species' current distribution with their past distribution, and how the planet Earth changed, like the tectonic of plates and the formation of the Isthmus of Panama, and when the Amazon River started to flow towards the Atlantic, and then there was a biogeographic different scenario for species occurring there. And then we can start to understand the relation of the species with the environment. So we can understand, for example, these stingrays, they are adapted to salty water. So when the Amazon freshwater started to flow towards the Atlantic, there was a huge increase in freshwater and sediments, nutrients, and it was no longer a good environment for these stingrays to live, millions of years ago, and they ran away from this. But we can also see it now. For example, there is another stingray that occurs only in northeastern Brazil. It's endemic, and it occurs only in salty water and water with high temperatures. And probably it's related to this time when the Amazon started to flow, because this stingray ran away from the Amazon outflow. So it's interesting, we can relate past events with current species adaptations. So we can do that using genomics. That's how we can do it. They changed the way we can do things nowadays. So, for example, the environmental DNA, which is something that people are still trying to improve the methodology. It's already amazing, but they are trying to improve it more. So you can sample some drops of water from the ocean, and then you can try to identify which species live there based on the DNA that you find on that drop of water. So it's fantastic. You can try to understand species distribution based on drops of ocean water. Yeah. It's fantastic. We are doing another research on race skates from south of Brazil and Argentina. And we're using another methodology, which is not genetics, it's ecological niche modeling, but we are trying to project how the ecological niche of these species might change in the future with climate change. So like they're currently adapted to colder waters, being close to the coast, but with climate change, like the temperature of the water is going to change, of course, but also the nutrients. So nitrogen is going to change, silicate, calcium, all the nutrients that are dissolved in the water, they're going to change their concentrations. So it's going to affect those kates. And we can see that their distribution is going to shift a little bit. It's going to shift not only southwards because the climate's going to be warmer, so they're going to be south. And they're going to be a little bit further from the coast, looking for deeper waters, so they can adapt to new environments, but not that new. They're going to look for environment in the future that's somewhat similar to where they are today.
Of course.
I'm going to talk about Argentina because I've been living here since the beginning of the pandemics. So I didn't leave the pandemics in Brazil. So here in Argentina, I've been working from home since the beginning. So I'm doing my postdoc from home, and I wasn't able to go to many places and talk to fishermen and go to the lab. I mean, I've been to the lab three times in 1 year. So the pandemics has, stopped me from going to the field and from having contact to other researchers in person. But on the other hand, it allowed me to have contact with people from all over the world through virtual meetings and conferences. So I have been taking part in a lot of conferences that I wouldn't have been able to go if they were in person, because they would be in Europe and it's too expensive to travel. But now virtually, it's possible. It's way cheaper. So this thing is better. And I'm also using my computer to do all of the analysis and contacting people. So there are pros and mostly cons. Well, of course, it's a pandemic. But if we look at something that improved a little bit, was the contact with other people. And now I think the virtuality is improving and being more accepted. So in the past, we could have done everything virtually, but it wasn't accepted. It wasn't acceptable. People prefer to do everything in person. And now they're, okay with the idea of doing everything online. The first step is to get to know the biodiversity. This is it's not applied, it's basic science, but we need to know what's out there. That's what I said about the genus of stingrays that I'm working on. There are 9 species that we are aware of, but after genetic studies, we found out there are 14. So first thing, we need to know which species are there. This is the first step. So keep doing this kind of job. And then trying to reach out to scientists that are doing applied science to do something related to conservation if the person is not an expert in conservation. So reach out to other specialists and try to propose some measurements. Try to, if the person is able to go and talk to the fishermen in the case of marine species and try to teach them, not only just say, no, you can't, but, you can shift from this to that. You can do this instead of that. And explain why and how. Leave the walls of academia and go to the community and talk to the public. Talk about what we do, why we do it, how we do it, in simple words. It doesn't need to be very complex, just for people to know what we're doing inside a university, because they just see it from the outside and they have no idea what's been going on there. And maybe we are describing new species and proposing conservation measurements and people are not aware of it. So we need to let them know. So these would be my advice, my recommendations for researchers and people that are interested in working on it.